Hi everyone, and welcome back to the next review video for the Biology or Living Environments regions. This is Unit 2, Topic 2. So the first thing in this topic is looking at the cell membrane. Our first question is, what is the cell membrane? If we remember from the lesson on organelles and looking before, the cell membrane, otherwise called the phospholipid bilayer, controls what enters and exits the cell. So the cell membrane is the barrier between the intracellular region, which contains the cytoplasm and all the organelles, and all of the extracellular region, which is everything that's outside of the cell. So the other key thing you want to remember about the cell membrane is that it is selectively permeable, which means that it selectively chooses what substances can enter and leave the cell. What it does is that it allows small and nonpolar molecules to enter and exit the cell, which are small to go through the phospholipid bilayer, while large and charged or polar molecules cannot just diffuse through. So now we're going to look at the parts of the cell membrane. The first thing we have is on the very outside are the hydrophilic phosphate heads. The word hydro, the part hydro that means that it's water related, and philic is attracted to. So these phosphate heads like water. They'll position themselves on the outside because we can see with just the diagram here of the bilayer is that it has two copies of these right here. On the outside, this letter D is the hydrophilic phosphate heads, which are positioned always on the outside. So this right here is the, inside in the cell, and this is out. Outside of the cell, it's exposed to fluid, which is why it's on the outside, and inside of the cell, it's exposed to cytoplasm, which is why they're always facing outwards. So the second part we have are the lipid part of the phospholipid bilayer, which are the hydrophobic lipid tails. So again, hydro is water, but this phobic is, as you know, like with phobias, it's afraid of water. So these hydrophobic tails do not like water or liquids, which is why they position themselves on the inside. So as we can see here, going back to the diagram, letter C represents the hydrophobic tails, and the tails position themselves inward. So you have the two tails on each side positioned towards each other, moving away from water. And then you also have proteins. So you have receptor proteins, which are found on the outside, on the outside of the cell, which are represented here by letter A. These can be either going throughout the entire. These can go out throughout the entire protein, or it can be just found on the surface. But these have specific functions, which we'll talk about in a second. And the second one are transport proteins, which are represented by letter B. And these ones have a role of helping with the transport of protein, the transport of material. But we're going to look at the proteins more deeply, actually, right now. So this is the next part, looking at the type of proteins. And our first type of proteins are the receptor proteins we mentioned above. So receptor proteins, their role is to receive messages from outside of the cell. The receptor proteins, when cells need to communicate with each other, they send signals that are received by receptor proteins, which receive them from outside the cell and can communicate to other things inside the cell. Next, we have the transport proteins, which go throughout the cell, and these have a role of helping substances move through the membrane. So if you need to get a large molecule in, something that's too large to fit through through the membrane, but you need to get it in, or it's a charged substance or a polar substance that would not regularly go through, you would go through a transport protein, which helps to move these specific substances through the membrane. And now the final one you have are surface proteins, which act really as a name tag. These are a marker to help identify the cell. So when other cells are communicating, they look at these surface proteins, and these surface proteins send specific signals to help identify each cell, just like a name tag. So those are the parts of the cell membrane, and now we're going to look at modes of transport. So transport is when substances are moved through the cell membrane, and there are multiple different types. So the first is active transport. And active transport, the key thing you want to remember about active transport is that it uses energy, or ATP, because we know that ATP is where we get our energy from. This is when cells are moved across it, and it needs to have energy used. And the other thing about this is that it moves against the concentration gradient. So it goes from a low to a high concentration. This means that, let's say there's a lot of a specific substance inside the cell and a little bit outside of it. If you want to get more substance into the cell, you would need active transport to push it against the gradient about where it would normally want to go. And to do that, you need to use ATP, or energy. Now, here we just quickly have the two different types of ways molecules can move. You have endocytosis, think endo is in, where molecules move into the cell. So endo you can help remember with in. And then the second one you have is exocytosis, and exo means out. So you have molecules that move out of the cell. This is just a key thing to remember. It really just helps to remember the prefix. Exo is out, endo is in, which helps you remember where the molecules are moving. So next we're going to look at passive transport. And passive transport is moving across the cell membrane without using energy. So no energy is needed for passive transport, which is one of the things that differentiates from active transport. But the key thing is that it moves from a high to a low concentration, which means that it's moving along the concentration gradient. The reason this has no energy is because things naturally want to move along the concentration gradient. If something's at a high concentration somewhere else, it will want to move towards that lower concentration. And this moves towards equilibrium. So what that means is that it wants to reach equal concentrations. So equilibrium is when there's the same amount of the substrate or whatever substance there is on both sides, and that is why they naturally go from high to low concentrations. So they can try to have equal concentrations. So now there are three types of diffusion that you want to know. The first is simple diffusion, which is just movement from high to low that goes through the membrane. So this is when small molecules, you can think like really small gases, they move straight through the phospholipid bilayer. You don't need a protein, you don't need anything else, no energy. It just simply moves through the membrane. The second one is facilitated diffusion, and this happens through a transport protein. So facilitated diffusion occurs through the membrane with the help of a transport protein. The reason why this is still diffusion is because it's still moving from a high to a low concentration, so it's moving along the gradient, but it might be too large to move strictly through the membrane, or it might be charged, which means that it needs the help of a protein to get through. And now the final one you want to know is osmosis, which is specifically the movement of water from a high to a low concentration. So osmosis is the movement of water, and one key thing which often trips people up with osmosis is that even if a cell is placed in salt water, the salt or the solute does not diffuse into the cell. The solute or the salt will never be moving into the cell. With osmosis, only the water is moving. So if you have a high concentration of salt, then the water will move towards that high concentration of salt or whatever other solute, but the salt itself does not move because it's a charged atom. Instead, only the water moves. So you just want to remember that the salt does not diffuse. So now, looking down, we have one of our key labs that you need to know, which is the New York State Diffusion Lab. And we have an image here which shows the before and after. So in the image, we have the start with our container. We have our beaker here, which has we can look at the key to see what they mean. We have starch and glucose starting inside our cell, and then an iodine, which is the starch indicator, on the outside. And then after leaving this for a while, we see that the glucose has diffused out of the cell, which is towards equilibrium. So there are an equal number of glucoses in and out of the cell. You can see there are 3 in and 3 out. The iodine has also diffused in, so there's an equal amount of iodine in and out of the cell, as you can see with 2 and 2, but the starch did not diffuse out. And the reason for that is because the starch is too large. So here we'll take some notes on what occurred in this lab and the key things you want to remember. The first is that starch stays inside the cell, and the starch cannot move because it's too large. It's too big to move through the membrane. The membrane in this instance, or the cell, is very fine, and the starch is too large, and there are no proteins to help it move through, so the starch will be staying inside the protein. However, the iodine, what we see represented as I in the diagram, which is an indicator for starch, turns blue and black inside of the cell only. This is because, since we know that starch stays inside the cell, and iodine tests for starch, while iodine is both inside the cell and outside of the cell, it can only turn blue or black when it's in the presence of starch, which occurs inside the cell. So the iodine inside the cell will turn blue and black, but the iodine outside remains its normal color. And then the final thing you want to know is that the Benedict solution, which is the indicator for glucose, will turn orange-red, which is when heated, which is the positive result when heated with glucose, showing that it's glucose diffused out of the cell. So it turns both inside the cell and outside of the cell. And this we can also see above on the diagram because we know that glucose remain in the cell but also diffuse out of the cell towards equilibrium. So the Benedict solution will test positive for glucose in both because the Benedict solution is small enough to diffuse through both, and glucose can diffuse through the membrane. But iodine solution only tests positive for starch inside the membrane because the starch was too large to diffuse out. So this is a key lab you want to remember, and often they will give you this exact diagram we had right up here, and you'll be asked to analyze it or draw some conclusions from it. So these are the key things you want to remember. Okay, so here we have some review questions. We start with number 8, which says, the diagram below represents a cell and some molecules in its environment. Which molecule would require the use of energy to be brought into the cell? Because it says requires the use of energy, we know that we're dealing with active transport, which will take us from a low concentration to a high concentration. So we want to see which one to be brought into the cell has a low concentration outside of the cell and a high concentration relatively inside the cell. So first we'll start with number 1, which you see are these little squares. We have 1, 2, 3, 4, 5 squares outside and 3 squares inside. So this means that it has high outside and low inside. So this would not be our answer, because this would be regular diffusion going from a high to a low. So our answer, I'll just erase that. Our answer would not be number 1. The next one, number 2, we have these little diamonds. We see 1, 2, 3 diamonds outside and 2 diamonds inside. So this could be our answer, because there are slightly higher on the outside than inside, but because there are only 5 total, this is functionally at equilibrium. So we'll come back to 2 later, but this is most likely not going to be our answer. The next one we have is 3, which are these little triangles. We have 1, 2, 3, 4, 5 triangles outside and 1 triangle inside, which means there's definitely a much higher concentration on the outside and a lower one on the inside. So it would definitely naturally diffuse in, which means no energy is needed, and number 3 is definitely not our answer. And now, finally, we're going to look at number 4, which are these circles. We see 1, 2, 3, 4 circles on the outside, and 1, 2, 3, 4, 5, 6, 7 circles on the inside, which means that there's a significantly higher portion on the inside and a lower on the outside, which means that in order to go into the cell, that would definitely require energy to go low to high. So number 4 would be our best answer, and better than number 2, because while there is one more on the inside than the outside for number 2, it's functionally at equilibrium, whereas there's a larger difference for number 4. So number 4 would be our best answer for this question. The next one we have number 1, the very first question of one of these tests, says a function of the cell membrane in humans is the 1, synthesis of amino acids. We know that's not true. That's the function of the ribosomes. Number 2 is the production of energy. That's not true. That occurs with cell respiration in the mitochondria. Number 3 is replication of genetic material, which we know is also not true, because that replication occurs in the nucleus with DNA transcription. And finally, we have recognition of certain chemicals. So we know that this would be our answer, because if we remember, our receptor proteins have the role of recognizing chemical signals from others. So the best answer to this question would be number 4. And now we have one last review question here, which comes directly from our New York State lab. We see the initial state diagram below with our key here, and then we have a question: which diagram indicates areas of these substances would be located after 20 minutes? So we have starch, glucose, and iodine. So if we remember, they all want to go to equilibrium. Iodine and glucose are enough to diffuse through the membrane, so glucose would be evenly inside and outside. Iodine would be evenly inside and outside, but because starch is too large, starch would all stay inside. So here we see starch is inside, which is, for number 1, starch is inside, which is good, but glucose is still inside. It hasn't diffused out, so number 1 would not be our answer. Number 2, we see starch diffused outside, so this would automatically not be our answer, because starch cannot diffuse. For this next one, we see the starch stayed inside, which is good. There's an equilibrium of iodine and an equilibrium of glucose. So number 3 looks like our best answer, but just to be safe, we'll check number 4. And this one, we see that everything went inside the cell, which is just definitely not true, because both this glucose and iodine would want to be at equilibrium. So our best answer for this is number 3. And that's it.